Good afternoon. My name is Sandy Markey, and I'm representing the Proteomics Interest Group at NIH. And in that capacity, I'd like to introduce today's speaker, Professor Carol Robinson. She began her career in the pharmaceutical industry in 1972 before turning to studies in chemistry at Canterbury College of Technology and the Medway College of Technology, where she won a final year prize as a graduate of the Royal Society of Chemistry. In 1980, she earned her Master of Science from the University of Wales under the supervision of one of the giants in organic mass spectrometry, Professor John Biemann. She earned her PhD from the University of Cambridge with Professor Dudley Williams, another of the giants, and after 1 year of postdoctoral work, took an 8-year break for the birth of 3 children. In 1991, she attended the University of Kiel to earn a postgraduate diploma in information technology before continuing postdoctoral fellowships at Oxford. In 1999, she was named a titular professor at Oxford, and in 2001, a professor of mass spectrometry at the University of Cambridge. In 2009, just in December, she returned to the University of Oxford as the Royal Society Research Professor and Dr. Lee, Professor of Physical and Theoretical Chemistry. Dr. Robinson is a recipient of many prestigious awards, including the Biemann Medal of the American Society for Mass Spectrometry and the Rosalind Franklin Award of the Royal Society. In 2004, Professor Robinson became a Fellow of the Royal Society and is the 2010 recipient of the Prelog Medal. As we'll hear today, her research interests are in areas of mass spectrometry, protein folding, and assembly of protein complexes. Just to remind you that following her lecture, we can join her for an informal reception in the library right across the hall. Please join me now in welcoming Dr. Carol Robinson.
Thank you very much for the invitation to speak here and the kind introduction. It's a real pleasure to visit. And what I would like to do today is to show you how we can use mass spectrometry in a very different way, perhaps, to what you're used to. You may have gathered from my title that mass spectrometry was first used to analyze rare gases, and I want to show you the sort of huge leaps that it's taken since its discovery to today, whereby we can interrogate intact ribosomes and viruses, and I think this is a very exciting transformation. So if you think of a very crowded cell, you'll know that proteins that are packed very densely in the milieu of the cellular environment. And really the question is, and this is going to be a sort of recurrent theme throughout my talk, is what happens to them as you take them from here into solution and then into the gas phase. And imagine the gas phase is a very new environment for structural biology, and it's not clear what happens to protein complexes once you take them out of an environment where they're happy in the cell, through to water, where we know that there's many ambiguities about getting the right type of solution phase, but then into the gas phase, where there's a huge number of degrees of freedom for them to explore what really happens to their structure. This is my favorite picture to really, I think it really describes what we're doing here. So we're taking something that's very happy in the water, and we're trying to maintain its interactions, but get it to fly through the mass spectrometer. So it's a phase change in which we change the properties, but hopefully not the interactions of the molecules of interest. So this was ahead of its time. And I'm going to give you a little bit of history as to how I got into this technique, because it's not really obvious that you would come to a mass spectrometer and start to fly huge ribosomal complexes. So what were the steps? What were the transitions that made me and others think that this was an interesting and worthwhile thing to do? So when I started as a postdoc at the University of Oxford, we were very interested in protein folding, and particularly the effect of molecular chaperones on protein folding. This is a very early slide. It's very low tech, you can see. We didn't know the molecular structure of the GROEL chaperone, which of course is now solved, but at the time in 1994 we just represented the 14 subunits of GROEL as these yellow blobs, and we were interested to see if we could take a protein ligand which we deuterated. So with mass spectrometry we're measuring only mass to charge changes, so we have to, if we want to look at changes in folding, we have to label with a mass-sensitive label, so we use hydrogen-deuterium exchange, and we can measure changes in folding in terms of changes in mass. So we deuterate our ligand, form the protein complex, so this actually goes into the center of the GROEL cavity, and then label this in water, and then watch and wait to see how folding is affected within this chaperone. And this was a sort of typical spectrum. So this is a mass to charge scale here, and this is intensity. And this tells us that we have many, many charges on this protein complex. And this comes about because lots of different protons stick onto the outside of the complex. It falls apart. So this is just a single subunit of GROEL. And these additional peaks that I've labeled with a red star come from the protein ligand right within the center of the cavity. So the question was, how does it fold within the cavity? We were able to address this and to monitor it with time, and we could see that it was partially folded. But if you think about it, electrospray ionization was only recently invented when we did this. It was discovered and published in the very early 90s. And nobody really understood the mechanism, so it wasn't clear if this was actually a valid experiment, because it could be that as soon as we applied the voltage, the complex fell apart, in which case exchange could take place, or it could be that the whole complex was transported into the gas phase. This is what we wanted, and then we used the gas phase to break it apart, so exchange just stopped because it's in the gas phase. And the reviewers of the paper suggested that we tried to prove that this complex was actually intact in the gas phase. And you can see that this is in 1994, and this was actually quite a lengthy experiment, because in 1999 we finally succeeded in producing this, which by today's standard is quite ugly, but it showed us that we could in fact maintain the 14 subunits of GROEL inside the mass spectrometer. So they would go from solution into the gas phase, and instead of all falling apart into 14 subunits, we'd kept them together. We had a species now which measured 800 kilodaltons, and I like to think that this was actually a single ring version of the double ring complex. But today, this is how it would look, much, much clearer. And you can see, obviously now there are crystal structures from Art Horwich's lab, and Paul Siegler, actually, sorry. This intact 14mer has a really sharp, defined peak here, which means that there's 67 protons all over the surface here, and that rather ugly lump turned out to be the 7mer, which actually then proves that there were two rings. Now you can imagine, by the time we get this spectrum, very few people are really interested, because this is now a really well-characterized complex. We know it's two rings, it's got a fine X-ray structure. This is just mass measurement. But I think why I was excited was because I thought, when we don't have a crystal structure, when we have things that are impossible to study in such detail by conventional structural biology, we could start to make predictions. We could say, yes, it's a 14mer, it's composed of two rings, two 7mers, etc., etc. So I'm not going to dwell on this in detail, but I'm just going to show you a little bit about how the mass spectrometer that we developed actually works, and then move on to some exciting examples, hopefully. So this is all of the different parts that I'm going to focus on. First of all, we introduce the sample through a capillary, which we draw. We pull these in our laboratory. Typically, we have 1 to 2 microliters of an aqueous buffered solution. Clearly, we don't want to denature anything. We want to keep everything together. We want to keep everything at low pH. Mass spectrometers typically operate with acid, organic phases, high temperatures to desolvate droplets. What we want to do is keep everything as cold as possible, as gentle, and least perturbing. So we put as low a voltage as we can to induce an electrospray. We have many, many droplets forming. We want these to be as small as possible so that our desolvation events are very, very gentle. We gently evaporate away the solvent and get what we think is a naked ion, which actually resembles what it was like in solution before. Why do we use nanoflow? So this is a special type of electrospray which has a very, very low flow rate and a very small droplet size. And I just want to compare it with the conventional electrospray. And you can see here, this is Growe-L-Fortima. This is Growe-L-Fortima in conventional electrospray, which has a much broader bore, much larger droplet, and you can see lots of aggregation, and it's actually then much more difficult to determine the stoichiometry. Why is this? Well, we know that if we calculate from a certain concentration the number of proteins that should be in the droplet, if we have 180 nanometers, you can see there's many, many. And if we have only 18 nanometers, if we do a Monte Carlo simulation of a particular concentration, we can find that we see 1, none, 2, or 3. And if we work this out graphically, if we take a 2 micromolar concentration, which is where we want to be, most of our droplets will be empty, but there's just a very small fraction which has just 1 molecule of the protein complex. So the opportunity for aggregation is thus reduced. So where is the charge located? Now this is a key question, because you can imagine that if you're drying this complex down and there's huge numbers of charges, if they're in the center of the complex, it's going to be extremely disruptive for protein-protein interactions. And what we believe is that as we desolvate the ions, it's actually on the surface. What makes us think this, I hope you can see this is not the best of slides, but there's a linear relationship between the surface area of a globular protein complex and its charge. And there's not a linear relationship between the mass, which is in red, and charge. You can see that this is curved, but this is linear. So we're pretty confident that we can actually predict almost the surface area of a protein complex based on the charge that it has. So it's a really very tight relationship between surface area and charge, which is one of the things that leads us to believe that the charge is on the surface. A second experiment that also makes us believe this is that if we take another system, very like the Growe-L, where we put ligands down in the center, we find that with the proteasome, which acts to remove proteins targeted for degradation, if we unfold a protein such as cytochrome C and put it down in the center of the cavity, it has to be unfolded to get in, we actually can't see it because it's not charged. It's actually protected by being inside that cavity, so it's not an exposed surface area. Whereas if we don't unfold it, but we just stick it all over the surface of the proteasome, we get lots of non-specific aggregation in high concentrations of cytochrome C. We can actually now see it because it's exposed, it's on the surface, it's charging. So this leads us to believe that all of our charges are on the surface. So the next region to look at is the quadrupole. What happens here? Not in detail, but if you're taking through a whole mix of protein ions of different sizes, the quadrupole really acts as a mass filter. It's 4 rods, of which the opposite ones are paired, and RF is applied in pairs, and actually the ions sort of spin through here, and they're propelled forward by a DC voltage. for our experiments, we exchanged the typical 4,000 mass range, 4,000 mass to charge quad for a very much higher one. So we changed this for 32,000, which meant that intact ribosomes and viruses, etc., could be studied. In the collision cell, what we want to do here is really interrogate these complexes. So we want to activate them, see what comes off, see what components they have. And in terms of a protein complex, it's coming through here, it's being bombarded with lots of neutral gas atoms, which is activating it, and you can see that single subunits come away and they actually start to unfold. And I'll show you why we believe this to be the case in the next example. So this is a small heat shock protein, and it's a 20-former. This is its crystal structure. And if we isolate just a single charge state using our quadrupole mass filter, we just choose one particular peak, and then we're going to activate it, so we bombard it like crazy with lots of neutral gas. What happens, the 20-former becomes a 23, a 22, a 21, so we successively remove single subunits. But what's interesting about this process is that if we look at the intact 20-former, it has 47 charges. It loses one subunit. It has, on the remaining 23, it has 33 charges. So this one subunit that's come away has taken approximately a third of the charge with it, which is huge, which is why we believe that these proteins are actually unfolding, forming a sort of point source, which then funnels away the charge in this very asymmetric manner, because if you compare how much of mass is going away, it's about 4% of the mass, but it's a third of the charge, so it must have increased its surface area. So the charge partitioning is directly related to the surface area, and you can see, I think, that the surface area of this unfolded monomer is about 30% of this compact structure. So it's coming away, it's unfolding, it's acting as a point source of which charge is funneling down, and then the two charged particles dissociate in the gas phase. And this becomes important later when we start to look at complexes where we don't know the structure. We can start to make predictions based on what's coming away first. Why are certain subunits lost first? And this is something that I'm very interested in, because if you want to use it predictively, you have to understand the mechanism. And the first complex I'm going to show you is the intact proteasome. And exposure is very important here because, as you know, this has 2 alpha rings and 2 beta rings, and it's a sandwich. And what we do is we see loss of alpha subunit because it's exposed. You can't lose beta because it's sandwiched between the middle. But if we now look at the half proteasome, you can see that rather than losing the blue, we have a choice now between the blue and the green. And the green actually unfolds more easily than the blue, so the green comes away first. So exposure is very important. But we still haven't addressed the key question, and the one that's haunted me for many years since I've started in this area was, well, we know that charge is accumulated on the surface, but we're not sure what happens to them when we take them out of solution. So if we have a ring protein complex, is it still a ring when it's in the gas phase, or has it distorted, collapsed, turned inside out? Many theoretical studies suggested that actually they would turn inside out because the charge which is on the surface is solvated. When you take the solution away, it's likely that it's going to invert. And this would be a disaster for the future of my research, so I was hoping that that wouldn't be the case. But we didn't really have a way to determine this until more recently, and you may have heard of this. This is a different type of mass spectrometry, really the same overall mass spectrometer, but it contains just this new segment here, which is an ion mobility separator. And what happens here is you store your ions in this trap region, you release them, and then they go across in a sort of transverse wave motion, which really just gives them a longer path, and then you bombard them with inert gas. So if you're very large, in circumference, it's much harder for you to get through that. It's really just a sort of shape sorting device, and the very small compact ions of the same mass to charge will shuttle through much more quickly. So if we now take a really well-defined protein complex, in this case we're going to use the trap ring. This is an 11 subunit ring structure. 11 protein subunits, forms a perfectly beautiful symmetrical, well, not really symmetrical, but planar 11-membered ring. And you can see it has this spectrum here, so on average around 20 charges over the 11 subunits, so roughly 1 or 2 charges per subunit. And if we now look at the shapes, you can see that this red line actually equates to the ring structure. So this is now the collision cross-section. We've measured this going through the ion mobility chamber, and we've calculated its collision cross-section, assuming it's a ring. And this is what we saw. Perfect ring, slightly buckled ring, and collapsed. So this is for these 2 charge states here. So the more charge we put on, they start to collapse. At first that seems counterintuitive. More charged, you'd think they'd want to stay away from each other. But actually they're coming through at much higher energies when they're more highly charged, and this is causing them to collapse. But what was really exciting was that we then wrapped a piece of RNA around the outside of this ring, because it's an RNA binding protein, and bound the 11 tryptophans. So this is now the apo form of the complex. This is the holo form with the RNA and the tryptophans, 2 populations. The apo form collapses as it did before, but the holo form is now larger because it has the RNA contributing to its collision cross-section. And the thing that was exciting was that it didn't collapse. It was really quite a stable ring structure that wasn't distorted and wasn't collapsing. And so this really proved to us that although it's counterintuitive, you take away the water, these are not collapsing that readily. If you overcharge them, yes, they can collapse. If you activate them excessively, you can collapse them. But if you can maintain the right conditions, you can keep them in this ring shape. And at first I was a bit disappointed because we had apo and holo. I would have preferred to have a pure holo solution. But this was really its selling point, because if you imagine, if you take a complex from cells, it's very unlikely that you're going to get pure complex. And so actually being able to go through a mass spectrum and ask independently the shape of each of the components is a hugely powerful thing, because most SACs or similar techniques would average across these populations and come up with an average collision cross-section. So this was really, although somewhat messy spectrum, was actually turned out to be an interesting point. And so just one further thing before we start some unknown protein complexes. You'll be familiar with the problem, or not the problem, but the idea that protein complexes, which are either hexamers or 14mers, may have different subunit packing. If you're a crystallographer, you'll be very familiar with this. So, for example, 2 hexamers can be composed of 2 trimers or 3 dimers. So these red lines indicate the strength of interaction is in this direction or in this direction. And for this kinase and GROEL, we have 7 dimers or 2 7mers. We wanted to have ways of distinguishing these possibilities. And what we did to look at this was actually quite an interesting exercise. We went through the whole PDB, not me personally, but colleagues at the LMB, Sarah Teichmann and Emmanuel Levy, and they plotted and predicted the strength of interactions of all of the homomers in the PDB. And then we wrote to people around the world and said, please can we have some? And actually, most people were really quite obliging, and we got a fairly large return for our efforts. I'm not going to talk to you about all of them. We had about 40, I would say, in total. 40 complexes, and then we used partial denaturants to tease them apart. So in the top row, you can see that we've maintained the overall hexamers and 14mers, but in the bottom row, we have now a trimer and a monomer, because this is the strength here, a dimer and a monomer, because this is composed of 3 dimers, and then, of course, the heptamer of Groeal. So we reasoned that if we were given an unknown complex, we would not be doing this. This is the realm of crystallographers, but we might be doing this, because we can now tell you that it's composed of trimers or dimers, etc., 3 dimers or trimers, or planar rings, etc., etc. So we've always been able to do size, because that's what mass spectrometry does. Stoichiometry you get from size. Symmetry and shape are the 2 new S's that we can now add. So symmetry of packing and shape from this ion mobility separation. So we're now in a situation where we could tackle some unknowns. Now, for mass spectrometry, if you think about this, actually homomeric complexes are not so interesting, because we're distinguishing things on the basis of mass. So if you have something which has all the same mass subunits, it's less interesting than if you have a heteromer, because then you can start to say who's next to who, which particular subunits form stable modules, etc., etc. You have a much greater opportunity of getting structural information if all of the subunits have very disparate masses. That's the ideal complex for a mass spectrometrist. So I'm now going to tell you, I've given you all the background to the developments and hopefully helped understand the technique. Now I'm going to apply it. And as you know, mass spectrometry is generally used for this type of network of interactions. Vast arrays of proteomics data can tell you or predict numbers of interactions, but what we wanted to do was actually pick them out and see if we could see them as intact complexes. And this is our first example of the yeast exosome, which I'll go through only briefly. This one I really like because it's such an odd-shaped complex. It's got 13 subunits. Ideal for mass spectrometry because it goes from the very small to the very large, so from 20 kilodaltons through to 140 odd. So you can really get this nice range of scale, and you can really work out who's next to who. This one I won't talk about today because there isn't time. And then for the second part of this applications, I'm going to concentrate on a new area for us, that of membrane protein complexes. So the very first one that we did was to fish out the intact yeast exosome. Now, the yeast exosome does not yet have a crystal structure, so this was one that we could do in advance. And this is how we build our model. This is the intact spectrum, which tells us that there are 10 protein subunits in the model. It's a 400 kilodalton complex. And actually, this still surprises me, because if you just took something out of a cell, even using tandem affinity purification, you might imagine it would look completely awful. I thought it would look terrible, but actually it's really quite homogeneous. We also found some very small components at low mass to charge, which when we interrogated them, turned out to contain some of the subunits we were looking for, either as dimers or trimers. So this is starting to give us building blocks from which we can build a structure. We then perturbed the complex by adding partial denaturant. We got very lucky move this was, because we then got 3 pairwise dimers, and we reasoned that we didn't really, it was sort of known that it was likely to be a ring structure. So if you have 3 pairwise dimers, you could argue that it's most likely a 6-membered ring. We then tried to arrange these by placing bridging subunits, which conferred stability to the dimers at the corners. And we also looked at neighboring losses. So if you lose something from one side of the ring, you can't lose something from the other side of the ring, because the ring falls apart. So if you lose one thing and you lose the neighboring thing, then they must be next to each other. So neighboring interactions. And actually, in all honesty, we could never really find a place for this very large subunit, but we knew that it made one side of the ring very much more stable than the other. So this was a very stable side of the ring, and there wasn't much space. This is not a good argument, but there wasn't anywhere really to put it except on the base of the ring. So this was kind of a lucky guess. And we made this 3-dimensional model from all of these building blocks. And you can imagine maybe you can't, but if you're a mass spectrometrist and you try and produce a three-dimensional model for the first time, nobody's really going to give you the time of day, and no, it can't be done because you're just measuring masses. How on earth could you come up with anything with any shape? But we were confident. But one of the difficulties we had was we had these pairwise dimers, these bridging subunits, but there was already a paper out there, and it was published in Science, in which all of the interactions had been predicted from databases. And our model, unfortunately, didn't agree with all but one of them. So you think of all the possibilities you could have, and we only had one in common, one pairwise interaction in common with this particular prediction. But actually, I have to say, the people who made the prediction at the EMBL were great, because we wrote to them and said, you know, we've got these interactions, it doesn't agree at all with what you're predicting. And they said, okay, we'll model yours. And then they said it actually modeled much more easily. And this was the now atomic structure model, not structure, model that we could come up with from homology from our interactions. And fortunately, this came out before the crystal structure of the human exosome. There's still no crystal structure of the yeast. So we still believe that this is correct, but of course there's nothing to prove this against, except by looking at the human, and ours is identical in terms of where the homologous subunits are interacting. And we can then also look at where the proteomics data fits on top of this. And I think this is very interesting, because it's really, once you have a structure, and you can have structural context then for your phosphorylations, you can see that they're all clustering in this corner, and that's because this complex then interacts with the ski complex, and so you can see phosphorylation is very important here in governing those interactions. So I think actually the future for me, and the excitement, is in taking structural complexes and then superimposing on the complexes the proteomics data. So we were encouraged by this because we'd got to a structure from masses. So we took on something much more challenging, which is this green structure. This is the 40S subunit of the human ribosome, and this is iris RNA in yellow here. We were interested in this green part. And we knew from proteomics 13 subunits, and this was its spectrum. Now this spectrum just looks like a typical GROEL spectrum, but it's actually so much harder to get. So we struggled for many months on this, and she turned out to be quite competitive, because it was sent around the world, this particular sample, to mass spectrometry manufacturers and to our lab. And this, thanks to a very talented postdoc in my group, she got this beautiful spectrum, which makes it look like it's a recombinant complex, but actually it's very heterogeneous, lots of post-translational modifications. And she got all of the subunits together at one time, just a small amount of subunit I, which is coming away. We then need to identify the subunits according to their masses. Now, this may sound trivial. You think, well, just look them up in the database, but actually they're nearly always wrong. So you have to go through this rather laborious process of getting a bit of sequence information and tying it together with the mass. So you have to separate them, work out their mass, and then digest them, make sure that what you think, because if you identify something from its mass and the mass is wrong, then the whole of the rest of your assignment is out the window, basically, because you just can't rely on it. And then what we did was to break this 13 subunit complex into smaller modules, building blocks for building the structure, and this is just one set of these. So we manipulate the solution conditions. In this case, it was the ionic strength. And you can see pairwise, two trimers, ELK and FHM. And these start to build into larger modules. How are we going to fit them into the EM density? Well, you remember I told you about the shape sorting ability earlier on. We now take the trimeric complexes, two trimers, FHM and ELK, and we can see if this is a linear array of subunits, it will have this green collision cross-section. If it's a closed packed, it'll be purple, and we can see that it's actually linear. And in the other case, FHM, This would be the linear array, and this is the closed packed. So we can not only make it into smaller and smaller parts, but we can then just deduce the shape and the arrangement of those subunits. And we know from the EM density, you can't really see this very well here, but this is actually like a linear arm that hooks from front to back. You can see there's a sort of hole through here, whereas this one is a very much more close-packed trimer, and we believe this is where this is, this has been antibody gold labeled to show that it's subunit A. The only ambiguity we have left is in these two limbs. We're currently trying to sort that out with more antibodies, but we believe that it's C and D like this. All of the rest, if you think about it, and I think about this often, but we have 27 subcomplexes, of which we know all of their components, and then we have, the way I used to do it was to try and build it by hand and sort of think about, oh yes, that must be there and this must be over here. But we then had to have a computer algorithm because it's really not possible to consider all conceivable differences just in your head. So we also use network algorithms now to predict the nearest neighbor interactions, and this is what we come up with. Superimposed again the proteomics information, all of the phosphorylation sites, and I think again this is very interesting because they're right in the core of the complex. They're affected by HCV iris binding, which is shown by this shadow. And actually recently I gave this talk and somebody said to me, oh, you know, these balls are all very appealing, but actually do they tell you anything at all about function? And it's an interesting question, but actually they do, because these two subunits here, this one, the grey one, J, is close to the decoding centre of the ribosome, and the two yellow subunits, which were not known to be interacting, are actually RNA recognition, contain RNA recognition motifs. And you can see that they then, because they're so close to J, you can see how it might orient the RNA close to the decoding centre. And we can also see that these modules contain very similar subunits. So these are metalloproteinase, these are PCI subunits, and these are structural subunits. So by just knowing the phosphorylation, the protein interaction pattern, and where things bind on the ribosome, we can start to make some predictions about function that we can then go on to test. Okay, so in the last half, quarter, I'm going to spend on membrane protein complexes. And always at the end of my talks, until very recently, a couple of years ago, I would get to the end and I've done all these soluble complexes, and people would say, oh, you know, that's really exciting, but why can't you do membrane complexes? Well, why can't we? And it was a big question, because if you think about it, membrane complexes are difficult because of solubility. If they're in the gas phase, why should it care? Once they're in there, it should be fine. You don't need to worry about having huge amounts of detergent. So this is typically what we would see. I don't want to make it sound too easy, so I'm going to show you all the terrible spectra on the way. So this is a terrible spectrum. And this, you can see, is an unresolved hump, which you wouldn't normally show to anybody because it's too ugly. But what this means is you've taken a micelle, which is a huge collection of detergent molecules arranged rather like this, with a protein in the center, or a protein complex. And what's happened is it's gone into the gas phase, and rather than being a defined entity, it started to evaporate, it started to coalesce. So you have anything from a few detergent molecules to several thousand detergent molecules. So you just end up with this hideous hump. And we saw many, 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 many humps for years and years. But we just didn't give up. From time to time, we would stop thinking this is a pointless waste of time. But eventually thought we might just get this. And one of our problems that we had was we kept thinking there's so much detergent there, it's really ruining everything we're trying to see. We'll get rid of more and more of the detergent. And of course, as soon as you start to do that, you're still in solution, the thing crashes out. So you block the capillary, nothing is seen as usual. But what we found was that if you go above the CMC, and this was a Jack's paper of a few years ago, we started to see that it wasn't quite so ugly, still quite ugly, but not quite as ugly as it was before. And that maybe we were getting a slightly more defined micelle. But typically, no matter which detergent we used, we had this heterogeneity. And sometimes we would see subunits from the soluble part, but never from the membrane part. So if we had a complex soluble membrane part, we'd see soluble bits because they'd stay in solution, but never the membrane bit. So we had this kind of dream that you'd take a complex, you'd have it in your micelle, and rather than this, which is what typically happened, so you'd put it into the gas phase, it would fall apart, detergent would stick all over the place, and unresolvable spectra. But we'd have this dream that the interactions would stay, but the detergent would evaporate as if by magic in the gas phase. We would bombard the complex, but at a certain energy whereby we weren't disrupting the interfaces, but we were dislodging the detergent. So could it happen? This was crystallized by Doug Reese's group. This is BTU-CD, which is a transporter, vitamin B12 transporter. This is the membrane-embedded 2 subunits, and this is the ones that are in the cytoplasm, so these are the soluble ones. And this is recombinant, so we thought we have a good chance here. And we put it into a DDM micelle. Still quite ugly here. This is because we've got too much detergent hanging around, and we've started to dislodge the soluble subunit, the orange one. If we get the conditions right, however, we found that we could really start to see this intact membrane complex. If we overshot the conditions, not only would we get rid of the detergent, but we'd also start to lose the transmembrane subunit. So it's a really kind of intricate balance, but we can make this look quite nice now. This is the tetramer, intact tetramer, 2 transmembrane, 2 soluble subunits, revealing its post-translational modifications and lipid binding, which is also interesting. So it's really the micelle itself that's protecting this. So you keep the micelle there, you transport it into the gas phase, and then you activate it away. And then we can do all sorts of things such as gas phase dissociation to look at various things. I won't go into that. It's a bit specialized. But the ATP binding was interesting because this is this yellow spots here. We could see that once we added ATP, it was very cooperative. We went from 0 to 2. There was no 1 ATP binding. There's populations with no ATP or 2 ATP. Was that just a fluke? For a long time we thought maybe it was a fluke because it had a his tag, which is very favorable, gives us a bit more charging. But then we looked at several other membrane complexes, some in which there were no structures. MAC-B, we weren't sure if it was a monomer or a dimer. We found that it was actually a dimer. And you can see both 1 and 2 lipids binding to the dimer but not to the monomer. And we can then, of course, characterize the lipids. And just to prove that we can also do it with heterodimers, so it doesn't have to be the same subunit, we could also see with another one which hadn't been crystallized and was unknown stoichiometry that we could get a heterodimer plus a different cardiolipin actually binding in the center of this one. And of course, we can do things like take things where there's homologous structures in the database, and we knew that this had a monomer and a trimer. MEX-B, it's close to ACRA-B, so we could then predict its structure. One of the things we found was that the conditions were very, very different because for the soluble complexes, the mass is along here, and the amount we activate them in the gas phase to get good spectra and keep them intact is plotted up here. So it's this line here. Traditionally, they all lie fairly close, but for membrane complexes, it's so much higher. And the reason for this is they're covered in this protective micelle, and this really proved to us that they were still in this micelle when they're in the gas phase because we have to activate them like crazy to get them out. One of the issues with that, though, is if you're activating them like crazy to get them out of the micelle, are they not destroyed by this process? It could be that they're now totally unfolded. So to probe this, we took a tetramer where we knew the structure. This is Kerbach 3.1. This is the tetramer, bit of monomer detergent and a bit of trimer forming. But we measure here its drift time. This is what ion mobility spectra look like. We measure the drift time, plot the collision cross-section, and we found that it actually matches extremely well. So this is 6,900 angstroms, and it should be 6,969. So that's within our error of our measurement, so we conclude that it's actually quite close to what we predict. This one, similarly, this is beta-UCD again, the tetramer and the measured tetramer, very close. What's interesting here, though, is there are differences in packing. For Kerbach, the 4 subunits each go into the membrane, so it has a different topology to beta-UCD, whereby you have 2 in the membrane and 2 outside. This is actually the trimer now that we're looking at, and you can see with the trimer, A, it forms more easily, and B, it starts to unfold because the collision cross-section gets larger, whereas for this one, it's tightly packed and there's no unfolding. And this is just showing what's happening here. This forms a trimer much more readily than this one because all 4 are very tightly packed in the interface. So not only can we keep their structure, we can also reveal something about their packing. And this is going to be my last example because it's my all-time favorite. And you may look at this and think, well, why are you even bothering? Because there's already a fantastic movie of what's going on in the ATP synthases. You'll recognize this. This is from John Walker's website. It's the mitochondrial ATP synthase, and it has a proton gradient across here, which drives this asymmetric rotor, which induces conformational changes in the soluble head, which synthesizes ATP from ADP. This is the stator. And as I say, you look at that and you think, well, you might as well not bother with mass spectrometry. How on earth are you going to make any impact? But if I talk to John, well, he wouldn't say there was anything wrong with it, clearly, but he would point out that this is composed from a mosaic of structures. So in order to get a structure for this rotor here, they have to disassemble the complex, they have to express the membrane subunit in the absence of all other subunits. So there's a question, is it actually like that in the intact complex? How many of these stators are actually present in the intact complex? Because they have structures of these stators, but they're not in situ, they're disassembled. And are there allosteric changes that are occurring between the head and the base? So if you have a method where you can look at it intact, this, I think, is very exciting because you can get stoichiometry changes in ligand binding, seeing how they change the structure, and also changes in lipid binding. So just very briefly, because I don't want to run out of time, this was my all-time favorite spectrum for a number of months. Had it pinned on my wall, because this is the intact ATP synthase from Firmus Thermophilus, and it's containing 29 subunits. But importantly, it also has this transmembrane region, which we couldn't see for years, and it comes out intact. And that's what's so nice about it, because this region is very hard to deduce the stoichiometry in the structure, because, as I say, it's often expressed recombinantly and allowed to assemble in a detergent micelle. And we also see some fragmentation, which I won't go into. What surprised me about these spectra, and this is the yeast, we can actually see ATP binding. I wouldn't have thought in such a very large and heterogeneous thing that we would ever see this. So I was quite surprised. You can see 1, 2, and 3 ATP molecules, or ADP. We can't distinguish that amount of detail, but we can see nucleotide binding, which is interesting mechanistically. But I'll just show very briefly what we've learnt. Firstly, a lot of the models contain only one stator, and from the mass and the fragmentation we know that many of them have 2, so they're actually much more firmly anchored. And we also know, this one was controversial, this is the Enterococcus hiri ATP synthase, and it wasn't really known how many subunits were in the membrane because the EM said 7, the X-ray of the reconstituted ring said 10. We said 10. The people who did the EM still didn't believe it was 10, so they then engineered a cysteine cross-linked version, and again it was 10. So they're now having to accept that it is actually 10, I believe. And we can also look at ATP binding conformational changes, but I don't really have time to go into those. What is interesting for me is the lipid binding and how this is affecting changes in packing. I'm not going to show you that one, it's too complicated. But this one is the E. hierae, and we find in this particular one, this breaks up differently. The membrane ring, which is very much larger, is much less stable. And we couldn't really work out why this was. But when we looked at it very carefully, we could see that the ring on its own, this was actually one of the embarrassments, because for a long time we thought, actually it's not 10, it's got 11 subunits in the ring, which was completely ridiculous because it didn't match anything that anybody else had done. And the reason we thought that was because it has massive lipid plug in the center, and the lipid had so much mass that we thought it was another subunit. But what we found was that we could actually dislodge the lipids, measure them, and we've done this now for both rotor rings. What's interesting about the thermos is that it has 6 very tightly binding lipids, but it's a 12-membered ring. Now, if you've got 6 very tightly binding lipids in a symmetrical 12-membered ring, it's quite hard to do that. So we've distorted this to give us a 6-fold symmetry, which was also seen in the EM, and then docked into that subunit C. But in this case, this is the 10K subunit from Enterococcus hierae. All this green here is the lipid density, and this was actually modeled in the original science paper, but it wasn't actually quite the correct lipids. The lipids are actually much larger. So what they did was to model the most prevalent lipid, but that actually wasn't the lipid that's present, and we found that it was this kind of double lipid which has 4 chains as opposed to the thermos one which has 2 chains. So it's really collected this lipid, as far as we can see, to make this sort of lipid brush in the center, which is changing the orifice of the ring such that it can then make contact with this blue subunit, which it has to turn. And without this massive lipid plug, this wouldn't happen. Then we're going to use ion mobility again to look at the structures of these parts. And I'm really just, although we can measure this, it's not fantastically interesting to get a collision cross-section of that, because it's, you're not really going to be able to constrain a model with a massive collision cross-section of the intact, even though it's nice from a technical viewpoint. It's completely useless experimental information. But what we can do is look at this membrane part, which of course there's no structure. We can see for C and L12, we can get a collision cross-section for this, but then we can look at the difference when we bind I. Now I, there's no crystal structure, there's a membrane and a soluble part, and it could be that the membrane bit is anchored and the soluble part is waving around, in which case we could see this with ion mobility. But what we find, and this is first of all showing you the 6-membered ring that we postulate from the lipid binding within the pairs, fitting it to the EM density, which has 6-fold symmetry. The 12-membered ring now changed into 6-fold symmetry, lipid binding within these pairs, only 6 lipids for the 12 subunits. Our whole complex now, and this is subunit I. And the reason we put subunit I like this is because the collision cross-section of ICL12 shows that this is actually quite compact. We modeled several different versions with I coming up in different angles, but our results are most consistent with this, and we actually have experimental evidence that shows that I actually makes contact with both status. So I'm going to summarize, and where do I think we've been and going? So when I started in Oxford the first time, which was in about 1994-5, the first complex we got was transthyretin, and this is a very trivial one for us now, but it was exciting at the time. So this is a recombinant complex, 56 kilodaltons, and we would expect that to fly every. At the time, it would be quite a battle to get that to fly. And then over the years, we were able to go through higher and higher masses, and of particular interest, I think, for me was the ribosome, because it's very asymmetric and we can look at subcomplexes. But more recently, I think the interest for me, or the excitement for me, has been in taking these very heterogeneous complexes with very disparate masses and trying to map them into EM density. And of course, a whole new world has opened up for me now that we can do these membrane protein complexes. And huge numbers of people to thank. This is my current group. This was our away day in my new Oxford College. Well, it's actually a very old college, but new to me. And a number of people to thank for the EIF3 project and the ATP synthase project. Without lots of input from instrument manufacturers, all projects are very difficult because you have to convince them that these are good ideas and they should really help to get them installed on your instrument when they don't really want to do this. And also, last but not least, Nelson, who has now left the group, but he was a very interesting postdoc from Chile who knew nothing about mass spectrometry, which was probably quite a good thing, but he knew everything about membrane protein complexes. He was really expert and fanatical about membrane protein complexes, and so he really helped us get this area going. And without him, we'd probably still be struggling. So I'm very grateful to him and his input. And thank you very much for your attention.
We have time for a few questions. If you would, come to the microphones. If not, let's thank the speaker once again, and I'd encourage you to come meet the speaker at the reception across the hall. Thank you very much.